Our next speaker is David Van Dyken.
Okay, great, thanks. I'll try and break that timeliness trend here. All right, so what I'm going to talk about today, here's a long title, here's a short title, is what I'm interested in understanding the evolution of gene expression noise. And so what I'm going to present is some theoretical work trying to make predictions about patterns of gene expression noise that we should see in nature. So what is gene expression noise? So gene expression noise is random variation in the abundance of mRNAs or proteins, either within a single cell over time, as these traces show, or among isogenic cells in a population. And actually, if we take measurements for a long enough time period, these two measurements are equivalent. So we can quantify the statistics with two basic parameters. One is the noise strength, which is measured as the Fano factor, which is the variance in abundance divided by the mean abundance, or the noise, which is the squared coefficient of variation, which is the variance divided by the mean squared. So these two measurements are going to be important throughout this talk. So the cause of gene expression noise is fairly well understood. It basically results from molecular randomness that causes these molecules to undergo random birth-death processes. But the causes of noise, or the consequences of noise, particularly the functional consequences, are much less well understood. One of the most commonly invoked consequences of noise is that noise causes random switching between states of a bistable switch. But most genes and most proteins are not involved in bistable switches, so we need to have a theory to understand what the consequences are for most of the genes in the genome in these cells. Okay, so this fits in with a lot of evolutionary theory that's been done about the evolution of robustness. So robustness is the maintenance of performance despite perturbation. We can sort of conceptualize this with a fitness landscape. So we have some genotype space, and a population undergoes a biased mutational random walk on this space. And what happens is the genotype will map to the phenotype. Now, if there is developmental noise or developmental perturbation, there is a one-to-many mapping of genotype to phenotype. So we have a phenotypic distribution that's generated by a single genotype. And we can call this imprecision. So the genotype has imprecise control over the cell state. Another form of perturbation is environmental perturbation, which creates uncertainty. So we may have a one-to-one mapping of genotype to phenotype, but now the environment is fluctuating, okay? So we can put these two together, and we have evolution that happens with imprecision and uncertainty. And this is what most organisms in nature face, and we're tasked with understanding how natural selection works here. So one of the issues with selection in this case is that genotypes, the phenotype expressed by a genotype is not expressed by every single cell. So the phenotype is actually a distribution over a population of cells. So understanding how evolution operates to select among these different genotypes is something that we're interested in. So this is all the abstract stuff. So what we want to do is we want to create a theory where we can actually map this and parameterize it with actual data. So what we need is we need a system where we can quantify the distribution of phenotypes, then quantify how the phenotype maps to fitness, and then we can understand how robustness is selected for in this case. Okay? So what we need is a genotype to phenotype to fitness map. The genotype to phenotype will be affected by gene expression noise, and then the phenotype to fitness map can be affected by random perturbations in the environment. So in our model, the phenotype is going to be the gene expression state of a cell. So this is the relative abundance of RNA and protein, and then within the proteome, how the proteome is allocated to resource acquisition versus biomass synthesis, and so on. And we can map the gene expression state of the cell to the growth rate of the cell population. And we're going to use this as our measure of fitness, which is a decent measure of fitness in many cases in nature, especially when populations are perturbed from demographic equilibrium and cells have to divide exponentially back to carrying capacity. Okay. So again, our genotype to phenotype map is going to be affected by gene expression noise, and we're going to try and understand the effect of this noise on selection. But in order to understand the mapping of phenotype onto fitness, we have to understand something about the effect of noise on biochemical reactions. Okay? So this is the classic Michaelis-Menten-Haldane-Briggs reaction. So it's a non-cooperative bimolecular reaction. So you have a substrate molecule that collides with a catalyst reversibly to form a product. And so empirically, in vitro, this is what you measure, right? So as the substrate concentration increases, the rate of the reaction forms this rectangular hyperbola. So the rate is saturating with the substrate concentration, okay? So there's a nonlinear mapping of the substrate to the rate, all right? So most reactions in the cell obey this type of kinetics, including parts of gene expression, specifically translation. Okay, and we'll get back to this in a second here. But what we need to understand first to sort of conceptualize how this nonlinearity is going to affect fitness is how noise affects nonlinear functions. So here's a hyperbolic function. Imagine that we have two different cells or two different points that have different levels of x. So that causes them to have different levels of some function of x. So the average here, so that's the average of the x variable. The average of the function is this white dot here, okay? Whereas the function of the average is higher than that, okay? So this is Jensen's inequality, which is familiar to most of us. Now what happens when you reduce the variance? So we can lower the variance between these 2 points, and what happens is that the average rate, or the average function increases. So what this means is that noise in the substrate will slow biochemical reactions. Okay, so that's the prediction. So what we need is a stochastic model that incorporates this nonlinearity. Most stochastic models assume linearity for technical reasons, but we won't assume that. And so we can construct a stochastic model that includes this nonlinearity, and we can compare it with stochastic Monte Carlo simulations to see how well it fits. So here's a simulation where we have some randomly varying substrate, and down here we show the accumulation of product over time, and each one of these gray lines is a realization of the stochastic process. So this thick black line is the noise-free or deterministic expectation. All of the realizations, or most of the realizations, are below that. The thin black line is our theoretical prediction, which fits very well. So as we reduce the noise in the substrate, the deterministic and stochastic rates come to agree more and more, okay? And so then we can do this for a bunch of different conditions, and we can actually reconstruct a stochastic version of the Michaelis-Menten curve, and we see the same effect. So that the greater the noise in the substrate, the slower the reaction rate. Okay, fine. So what does this have to do with fitness? So what we can do is we can use the model, and we can construct a coarse-grained model of the cell, where we imagine that the cell takes up extracellular nutrients using uptake proteins, so permeases and other importers, to fill intracellular nutrient pools, which are then used for biomass synthesis via translation. And the output is the growth rate of the population, so the doubling time. So we can construct a large multivariate master equation with nonlinear transition rates, and then we can solve this with a continuous approximation, and we get something like this. So the average growth rate with noise in this system is the deterministic growth rate, this thing right here, then multiplied by this big term where we see that there are all these minus signs. So this is a fitness cost of different forms of noise. So we have the cost of gene expression noise. So mRNA noise, transcriptional noise, directly reduces fitness. We'll come back to this. Protein noise, specifically noise as the permease concentration fluctuates, reduces fitness. And extracellular noise reduces fitness. So this is the cost of imprecision that we were talking about, the gene expression noise, and the cost of uncertainty in the environment. And basically, in this case, it's just a linear combination of the three. And again, they're all proportional to the squared coefficient of variation. Okay, so we can understand this conceptually, graphically, by looking at the mapping of these different things onto the doubling time. These are all parameterized for E. coli. So mRNA, the permease concentration, and the limiting nutrient all have this hyperbolic mapping onto the growth rate. And so based off this nonlinear averaging, we should predict that all of these things should reduce fitness. Okay? All right. So one of the interesting things from this is that transcriptional noise directly reduces fitness. And this is just because what happens is the noise, the fluctuations in mRNA reduce the translation rate, which is sort of an interesting thing. And since this is well characterized, the distribution, at least in E. coli, we can actually make predictions about the cost of this and the strength of selection for noise amelioration. So this is the translation rate with infinite extracellular resources parameterized for E. coli, and this is the mRNA distribution from Taniguchi et al. And so using these parameters, what we can see is a 1-unit increase in the Fano factor of mRNA causes a loss in growth rate of 7.5 proteins per minute, 223 proteins per cell division, or about 1.5 times 10 to the negative 4 cells per generation, which is a selection coefficient that's basically on the order of codon bias. Okay? So given the effective population size of E. coli, this means that selection against this noise, or this jump in noise size, is about 10,000 times stronger than random drift. But now we can do the same thing for a larger cell. So this is Saccharomyces cerevisiae, and this is a hypothetical distribution of the mRNA noise that has the same Fano factor, the same noise strength as in E. coli. And what you can see is that on the scale of cerevisiae, the fluctuations are much smaller, right? And it turns out that we can quantify the effect of transcriptional noise again, and we see that now in cerevisiae, the cost of this noise is only 20 times stronger than drift. So selection to attenuate this noise is very, very weak. Okay. So what we can see here is that because the selection coefficient depends on the squared coefficient of variation, and the squared coefficient of variation depends on the cell volume, actually specifically the number of molecules involved in the reaction. So as the volume increases with a constant concentration, the same noise strength, or Fano factor, leads to a reduction in the square coefficient of variation, which is what's actually controlling fitness. Okay? So what this predicts is that larger cells are insulated, are more robust to these gene expression fluctuations than smaller cells. Okay? Okay. Okay, so I'll finish with this. So what we can do is, again, we can parameterize this model with data from these different species. And so the efficacy of selection, so NE times S is over here, right? And then down here is a hypothetical jump in the size of the noise caused by a mutation, okay? Here is the drift boundary. Below this, drift overwhelms selection. Above this line, selection is stronger than drift. This is for a mutation at a single locus in these species. All right, so what we can see is that a mutation at a single locus that increases transcriptional noise in these species can be strongly selected in E. coli, but not well selected at all in cerevisiae or slime molds. Now this is global mRNA noise, so the total transcriptome. Now the fluctuations in the total transcriptome can be selected against efficiently in yeast, but still not very efficiently in the slime mold. Okay. So finally, what we can do is we can actually make a prediction about how strength of selection should scale with cell volume. And quickly, I'll just leave you with this. What this is we have the cell volume here, and this is the noise strength, or the change in the noise strength at the drift barrier. Basically what we see is that for small cells, selection can strongly reduce noise, but for even moderately larger cells, selection is very weak at selecting against noise. Okay, so I'll leave you with that. And sorry. All right, conclusions. And there's the theory for those of you who are interested. Okay. 9 seconds for questions.